Everyone. Welcome back to another event of Aquatic and Fisheries Science Talks. And this is AFS Talk number 40. Lega is my very long time friend, I think from since 2008 when we started our PhD at University College Cork in Ireland. For those who don't know, Cork is a nice little city in the southeast coast of the Republic of Ireland. Dr. Lega continued to live and work at Cork and calls herself as a Corkonian. Um, Looking back at Dr. Lekha's achievements, she is a gold medalist from PSG College of Arts and Science in 2003 from the Department of Biochemistry. Dr. Lekha Menon Margassery started her career as a guest lecturer in the Department of Biotechnology at the Bharathiar University, Coimbatore. She became a biochemist specialist at the Jain Institute of Vocational and Advanced Studies, Bangalore in 2005. She then joined her husband in Cork in 2008. She then went on to pursue a PhD in microbiology at University College Cork, Ireland. She graduated in 2013 and worked since then. She was a postdoctoral researcher in the School of Food and Nutritional Sciences and then moved on to the School of Microbiology. She then worked as a research scientist in a company called Atrotech Limited. Due to her passion in teaching, she took up part-time role of a lab demonstrator at the School of Microbiology and exam invigilator at UCC. She was working at the School of Biochemistry and Cell Biology until October, and now currently she is a researcher at the School of Microbiology, UCC. She is the president of UCC Indian Alumni Community, board member of Alumni and Development Office, and also one of the prominent members of the Indian Community Support Group initiated by the Embassy of India, Dublin. She was also one of the 100 finalists to qualify the Mars One project, a one-way ticket to Mars. Besides her passion for research, she is very passionate about dance. She is the founder of an Indian dance school in Cork called Atma. The troupe performs at various events in Ireland. She volunteers and does a lot of fundraising events to help the community in need. She was the first Indian to stand for local elections in Cork. She says, I am an Indian at heart and Irish by soul. So today she'll be sharing her experience working on her PhD project. I welcome again Dr. Lekha, and the stage is yours.
Thanks, Deepak. That was a lovely introduction. And because Deepak knows me quite well, as he said, we are friends since 2008. First and foremost, I would like to thank the organizers of the AFS Talks and for inviting me to be a speaker. And the entire team, in fact, I think it takes a lot of effort to organize such seminar talks. And also to all the participants who have come here to listen to the talk. And I welcome you all. Um, and I don't want to go through my introduction because I think Deepak has told enough of it. And because I assume that this audience also have non-scientific audience, so I would be going in basics and I will tell about my research, what I did in my PhD long back. As Deepak mentioned, it was between 2008 and 2013, so it's quite a while back because I moved into a different field altogether. I mean, though it's microbiology, I moved into wastewater treatment now, so marine field is my passion though, but I probably would get back to it pretty fast. So I would like to share my screen. Okay, just give me a minute. Select a window.
You all right, Lekha?
No, I'm just not able to see the window which I want to share, which is strange because I can see it here.
Yeah.
Let me try sharing it again. So share screen, AFS Talks, yeah. Perfect. So can everyone see this screen?
Yeah,
Yeah,
yeah, we can.
perfect. Okay. Yeah, thank you. So today my topic, which I've selected, is blue biotechnology drugs from our oceans, and this is, uh, I would say, it comprises about 40 to 50% of my PhD work. So I'll make it very, brief, and so that I can convey the message what I wanted to convey. Uh, so microbiology is actually a vast area, and people do think, and they have questioned me as well, that okay, do you know, like just by looking into a microscope, do you know, just like that, can you spot which microorganism this is? No, I'm not that specialist in microbiology, because microbiology is divided into, it's a vast, it's a huge area, and you can find microbes which are used in applications in industry, in agriculture, in medicine, and very many areas. In industry, basically, the enzymes which are produced by microbes is used. That's the application what they use. And even in agriculture, you find a lot of different applications. In medicine, which I'll be concentrating more, is the development of drugs, about natural products and so on. So uh, just to tell you like how alarming the situation is, there are a lot of fungal and bacterial infections, like you can see in the picture. You can see how, you have skin infections, you have discoloration of the nails, so all, and also here you can see, some discoloration on the skin. These are all skin infections, eye infections. So it's caused by, sorry, various fungal and bacteria, and these are called as microbes. So even viruses, they are called microbes as well. They fall under the same term. So that's why I said this is a vast area. And my specialization is mainly on natural products more than of the synthesis or of new compounds. So it's mainly natural products as drugs. So what do you mean as natural products? So any chemical compounds produced by natural sources, which can be from plants, animals, or microbes such as bacteria, fungi, viruses. All those classify under the term as natural products, and they have a very important role in the treatment and prevention of diseases. And this actually came up quite, I would say, okay. It says host disable participant screen sharing. Okay. Okay. So they have an important role in the treatment and prevention of various diseases. And if you see recently, there were about the natural products have come quite popular between 1981, from 1981 and so on. And after 2000, actually, there is a halt. There's no major classes of antibiotics discovered, which I'll be talking in the coming slides. And there are about 49% of these are from natural products. So all the drugs, what you see in medicine, like 49% of it are from the natural products. So it can be from soil, it can be from plants, it can be bacteria from soil as well. It can be from a fungal from a soil sources. It can be from bacteria from oceans, which I'll be talking. That's my main focus today. It can be from plants as well. So those are all natural products. And of all the new chemical entities, as I said, 49% are natural products. Now, there are treatments available, like we have antimicrobials, some of the treatments. Now, this particular one is penicillin, which is quite a common antibiotic. So this was actually discovered from a fungus. Um, but we do have a problem here. So as you can see, a nice picture on the left-hand side of the screen, wherein a scientist is trying to, actually kill those microbes, but then they're talking to themselves. So bacteria do communicate. You have to know that, and they just say that I'm fed up with this guy. Let's become pathogenic, and that's what bacteria is becoming. It's actually quite alarming situation, And that is why we need drugs from different sources. We need to discover drugs from different sources. So what are the drawbacks of the existing drugs? They're quite expensive. Not everyone can afford some drugs, and some cause. Okay, I can't see my screen. Okay. Hmm. Something is there's some issue with this. I don't know. I can't see my screen at all.
Maybe we can restart the
Uh,
Google
it's okay.
Slides.
Yeah, maybe I'll just stop sharing. No, I can't see even the Google, so I'll probably stop sharing. Yeah. Sorry about this. I don't know what happened. All of a sudden, my just went blank. So let's see. Perfect. Is it okay?
Yes.
Yeah. Okay. So some of the antibiotics, they can cause severe side effects like renal damage. Now, for example, if you take, you know, um, let me let me tell you an example. If you take, um, what's the best thing which I can tell you? Okay, let me give you an example of an antibiotic called kanamycin. So if you take now, all these are all aminoglycoside class of antibiotics. So you have different classes of antibiotics. And some antibiotics like aminoglycosides, cephalosporins, amphotericin, vancomycin even, on long term, I'm not telling short, on long term, if you just keep on taking the antibiotics, it can cause kidney failure. And that's one of the side drawbacks. And the next one, which is quite alarming and which we all have to actually keep an eye on, is the drug resistant strains. So basically, most of the organism is becoming drug resistant. Now, what are drug resistant? So, for example, you have a staph infection, okay, which is it could be a skin infection or anything related to staph. And generally, with penicillin, it could have been treated. But now they have found that only 5% of the staph infections can be treated with penicillin because the Staph aureus has become resistant. So it's become so powerful. So the best example which I normally give for an audience like this would be if you have seen the Hulk movie, Eric Bana, who's a scientist, okay, who's a radioactive scientist. So if he has an exposure of gamma radiation, he becomes a Hulk and his personality changes. So basically, he's not normal anymore. It's the same thing with the bacteria. So what you give it, the bacteria has become so much like you overdose with antibiotics, it just changes over the course of action. So everything can change, humans can change. And what happens is it becomes resistant to that particular antibiotic, and that antibiotic will not work anymore. How much ever you give that, how much ever you give the patient that antibiotic, the infection remains because that particular bacteria has become resistant to that antibiotic. So we have to go and search for new antibiotics. So this is quite alarming. And a UK medical officer, previous UK, Sally Davis, she has said that we are in an arms race against these microbes. And I think we all should be careful. It's an equivalent problem, just like how we talk about climate change. Antimicrobial resistance is an alarming issue. And we all actually have to play an important role in this. And I normally take an opportunity to tell this that, for example, if you're getting common cold, don't take antibiotics. It's just a viral infection. Um, Even if your GP had like, okay, if you have a prescription saying, okay, it's not a common cold, the cold is caused by some other things, it's fine. But otherwise, just because you have antibiotic at your home, just don't take it. It's just a common cold. It's if you have an antibiotic, that's not going to kill the virus. It's just going to kill the normal bacteria which is present inside your human body. So I wouldn't suggest taking antibiotics just like that. So if we also create awareness in the community, I think we are equally responsible for the world to become like this. where there are a lot of drug resistant strains. So I think it's our responsibility as well to reduce it and also rediscover new compounds as well. As I know there are a lot of scientists in here, and I think it is our responsibility as well to go through a different source, find out compounds, discover new compounds from different source. And that's where my research comes in. So before going to my research, I would like to say that, as you can see, this is a timeline of when and where the antibiotics was discovered. So if you see after 2000, there's not much class of antibiotic discovered. It's come to a halt. So over these 20 years of time, bacteria and fungus, in fact, have become drug resistant. Like there's also a fungus called Candida, which is the second most important pathogen in the world, and that has developed resistant. Same thing, I think everyone would know about TB, tuberculosis. That's become drug resistant as well. So it's time that we start looking into different sources to get our antibiotics or antimicrobials in general. So that's why we need new drugs. So the source from where I get my bacteria they're called marine sponges. As you all know that 70% of the world is made up of oceans, and you have beautiful organisms under your ocean bed. You know, it's also undiscovered and it's not tapped much. Like in our Ireland itself, it was quite only in early 2000 that marine field has just shown its, has developed. So, and so we've actually, I'm telling on that note, I would like to say that we were one of the first batches in having this kind, having a PhD degree in this, because earlier, and we have our headquarters in Galway, wherein we store all these beautiful organisms which we collect, and mainly the collection of these sponges. And now it's not only sponges, my consortium was a wide variety. So we had people who were doing on coral reefs, seaweeds. So all these are beautiful marine organisms where you can, have develop natural products from. So I was working on sponges, and we had two ways to collect it. So if it was just a shallow water sponge, which is approximately 8 kilometers down the lakes, we used to have a specialist who used to do scuba diving, collect them, and then just on site, they used to just macerate it, and then so that we can get good amount of bacteria or fungus in it. The second way is we had PhD students who just used to go on a cruise ship, and they used to collect through robots. So the robot, you just used to go deep down around 1,000 kilometers depth, and they used to just collect the sponges. So these are the various ways wherein you can get the sponges. And they are called sessile filter feeders, and they're very simple organisms. So I'm going to show you a video of what I mean by sessile filter feeders. So for example, you will see in the video that two scientists, two scuba divers, who are marine scientists, they just inject an ink, and how, like a chimney, how a smoke comes from a chimney, you can see how, they just filter it, and you can see the dye coming out of the sponge. So I can show you the video of this. So
You might have to pull off the headphone.
yeah, sorry.
Yeah, it's not audible.
Coral reefs, sponges dominate the seafloor and the barrel sponges, rope sponges, encrusting sponges, and many other types. Sponges live from the frigid waters of the Arctic and Antarctic to the tropics. On many coral reefs, sponges dominate the seafloor and the drop-off. When they can feed, they do it by filtering tiny plankton from the water. A sponge is covered with small pores called ostia, which lead to a system of internal canals and eventually out to one or more larger holes called oscula. Within the canals of the sponge, chambers are lined with specialized cells called choanocytes or collar cells. The collar cells have a sticky funnel-shaped collar and a hair-like whip called a flagellum. The collar cells serve two purposes. First, they beat their flagella back and forth like fans to move water through the sponge. The water brings in nutrients and oxygen while it carries out waste and carbon dioxide. Second, the sticky collars of the collar cells pick up tiny bits of planktonic food brought in with the water. Sponges are very effective filter feeders since they're able to capture and eat particles as small as bacteria as well as larger particles. They might not look like they're doing much, but a simple demonstration shows how effectively sponges can pump water. On a reef in the Caribbean, I make a dive with a syringe filled with a non-toxic dye called fluorescence. By squirting it around the base of some sponges, we can observe how the water is moving by watching what the dye does. Within only seconds, the dye is pumped through the sponges along with the water. As you can see, a sponge is a pretty good water pump and also a good strainer. Any plant
So that's what I meant, that they are simple organisms and they're sessile filter feeders. So it is known that they filter around 24,000 liters per day. And in fact, the number of bacterial cells which they can have is quite a huge load. So even if you have one bacteria, coming in, though they filter, they'll be have, I mean, not one bacteria, I would say one time 10 to the power of 13. And if they just come in, you're going to have 100 millions of bacteria inside that sponge. And that's what we are targeting. So you can imagine how much of microbial load would be in the sponges. So they're not going to filter out everything. Some of them still reside, as you saw in the collar cells, which are present, that they would take them as food. So um, for example, I was working in a sponge called Haliclona simulans, and I had identified, we screen around 120 bacteria. So these are the different types of bacteria which you can see. So this is different bacteria. This is different. So like that, there was about 120 which we isolated. We can isolate more than that, but because I was running out of time, I need to start off my project. We started off with 120. So what are the functions? So what are symbionts? What is symbiotism? I think this is just like what to say. What do they do? So the bacteria, or the fungus, whatever resides, they are called symbionts. And something is wrong. Okay. So what happens is they have a lot of different functions to it. So basically, they kind of help in stabilizing the skeleton of them, because the skeleton is very important for the sponge, and also helps in the metabolic waste. And it also, the very important role, which is what the entire research was on, is to tap the secondary metabolites from the bacteria, which was produced by the bacteria, which has various functions, which can be antibacterial, antifungal, or cytotoxic effects. And one of the best example is salicylaldehyde. This is just a drug which was discovered by which was discovered or which was kind of found as a natural product in one of the marine bacteria. So as I said initially, that I was working on Haliclona simulans, and this is the bacteria which I focused. So why I chose that? So I'm just going to go through very basics of this. Now that's a media which is in which has starch, yeast, and peptone in it. It has some artificial seawater, and that's why it's called SYPSW media. But now most of the scientists here has adapted to marine with different variations of seawater in it, or artificial. You can even add some artificial, what you add to the aquarium. It's called instant ocean. You can add that as well. You can play around the composition of the media to see like at what percentage of the media you get good amount of secondary metabolite production. So that's all optimization. So I'm not going to go deep into it. So out of 125, we found 25 of them, which now what I mean by 25 of them positive and 4 strains positive with all these strains is like these 25 strains, they were active against all bacterial strains, pathogenic bacteria and pathogenic fungi. And then I have to narrow it down. I cannot work on so many strains. And so I focused my attention was on these 8 strains which I was working because it's easy always to get something working when you can get the product, when you can get the drug as a crude extract using chemical or using organic solvents. Otherwise, it's very hard if you go down this road. It will take ages because it could be a protein, but we didn't want that route, so we kind of stuck to this. So we found 8 of them, which is very active against pathogenic strains, which we tested in our lab. So this is what was done. So we had the, from the Haliclona, we got the bacteria, and then we cultured the bacteria for 7 to 14 days. So you can imagine how lengthy is the process just for the basics. We haven't even started checking out what antibiotic it is. We haven't even gone to that stage. It's just an initial process itself. It takes a while. And then we have to optimize the methods. We wanted to see how to get at least in a crude form. And so we did something like a methanol treatment and so on. And then we did we then started on what we get is very crude. It can have so many different compounds. We need to narrow it down to one specific antibiotic or one specific antifungal, which can be new or which we can pursue further, for the development, for getting a complete nice structure and to check its efficacy and so on. So it's like, for example, if you see, if I tell in a non-scientific term, I would say you have a recipe, okay? It has potatoes and onions and everything else. You have to focus on what you want, so you need your whole thing is your focus is your recipe, okay? So but in your recipe you have different components. It's the same thing. So you will have that's what is going to happen here as well. It happens always, and that's what you need a complete thing wherein you can just basically get to one thing wherein you can focus down. And for that, we call it purification. So I want to focus for an antimicrobial compound, which can be an antibiotic or an antifungal. And so that particular purification is done by HPLC. And this is like a phylogeny. So I want to know under which particular class is this SM8. Now, how do I explain the phylum? So for example, we are all in different communities. So we are all human beings, but then we have different communities. So we have Asian, we have Europeans, and so on. So that's the phylum for a bacteria. So SM8 is called in a phylum called Actinobacteria because it falls under further class called Streptomyces. So it has various divisions. A bacteria has various divisions. Its kingdom starts with kingdom, then phyla, then it goes to class, order, family, genus and species. So it falls under a genus called Streptomyces. Okay. Um, so coming to the, so next, now you got something crude. Now I want to know what is in that. So we use something called as HPLC, high performance liquid chromatography, and now it's kind of really advanced. So earlier, it's ultra performance now, so it's kind of very quick. But those days we didn't have that. We just had one analytical HPLC and prep HPLC um, in our lab. So I had to depend on someone, which I'll be explaining in the later stage. But this is what it does. You know, if you see the, I'm just going to play a small video, and this is what it does.
The Waters Auto Purification System for preparative scale chromatography. Preparative scale chromatography plays a critical role in applications where compounds must be synthesized, identified, isolated, purified, characterized, screened, and tested. Purification is used to isolate compounds for drug discovery experiments, to isolate unknown impurities or metabolites, and to purify standards, natural products, and biomolecules. Waters purification systems offer scalable prep configurations, whether you want the specificity of a mass-directed system or the inclusiveness of a UV system. The Waters Auto Purification System consists of a 2545 binary gradient module, an injector collector, the 2767 sample manager, a system fluidics organizer for automated column switching, and a 2998 photodiode array detector. Optional detection techniques can also be greater preparative flow rates to the preparative injector, on the flow rates to the analytical injector, and then much greater preparative flow rates to the preparative injector, on the same autosampler. An analytical evaluation of a crude sample is performed to determine if purification is necessary. A small amount of the sample is injected through the analytical valve of the autosampler. The sample is separated on the analytical column and sent to the detectors. When purifying the compound, a larger amount of the sample is injected through the preparative valve of the autosampler. The sample travels through the prep column and is split between the detectors and the fraction collector. Fraction collection software monitors the data and controls collection. In a system equipped only with UV detection, any peaks that achieve the required threshold are collected. When collection is triggered by the mass spectrometer, only the specific mass or masses of interest are collected. This increases the overall efficiency of the purification system.
So you saw how long it takes, you know, for a purification. So basically you have something which I've showed you in a previous slide in a bottle, which is a crude extract. And then if it's a smaller volume, just to see, just to test, just inject it using an injector, and then you just collect all the fractions, and then test it as well to see which fraction is active before even going to the preparative scale, which is a high scale, which is larger one. So it takes a while. It's not that easy to get, to discover new drugs. And this was in collaboration with University of Strathclyde. So what happened in University College Cork, that I was collecting I was cultivating the strain and then extracted the bioactive, and for the purification purpose, I had to send there. And then they used to collect all the fractions, send it to me. So there could be 120 or more fractions, and I have to test it against all the pathogenic strains to see which fraction was the best. And then I had to further purify it if I need to find a structure of a particular compound which of interest. So these are the steps that are involved when you say, okay, discover a compound. It's not that easy. It takes a while for a scientist to get to that stage, and at least those days here, because it was, I have to send it, back and forth. And I still remember my PI, my principal investigator, telling me, "Why are you concentrating just on one strain when you have 8 of them? Why are you, putting all the eggs in one basket?" And sometimes, gut feeling. That's what I told him. It's my gut feeling that it's the SM8 streptomyces, which has the best activity, and that's what I'm going to focus on. Because I think scientists should, I think they have something. I think it's there in every sense of, in everything actually, for any human being, that sometimes a gut feeling is right. And I was right. Like, I focused only on SM8. I'm not going to go in detail what I've been doing with it, but I can say you that we had identified several compounds, not into the structural stage, but we could say what it is. So we couldn't give it a name because we couldn't go to the NMR study due to lack of funding, because I was running out of funding as well. We call it fraction 17, so it's a saturated fatty acid, and it was an antibiotic, but we couldn't identify what the structure was. We could find something called as butanolites, and these, uh, I couldn't file a patent for this because of lack of funding again. But these butanolites, I have found a novel, I would say, activity. It has a novel application now. It's also an anti-calcineurin, which I'm not even going to touch because that's completely a different topic. Um, it also is an antifungal. You know, it can kill Candida, Glabrata, and albicans, and even an industrial strain as well, industrial strain of fungus. So it can kill that easily. Then we had certain antimycin, which is a known antifungal compound, and we found one antibacterial, which was in a mass of 227. But as I said, um, we couldn't go to the structural identification of it. Now, so as I said, those days genome sequencing was quite expensive. Like, it was 5,000 euros, and I had to pick one, and I picked SM8 for it because I found that it's got a lot of novel activity. So I wanted to see what is in the what is the genetic material of this SM8? Uh, why is it being showing a lot of activities? And so in, I think in December. So I was finishing my PhD in 2012. Yeah. So 2011 December, I extracted the DNA of this, sent it for sequencing. It was quite expensive, and those days only 454 was then. Nowadays, the latest technology, it's Illumina and PacBio and everything. It's quite cheap. Trust me. Now the students in the lab, they're just sequencing a genome for under $1,000, which is quite cheap. But I still got good amount of information in it. You don't have to see the numbers. It's just how much information I've got back. And this is deposited in, so we have a database called NCBI in science field, wherein you can deposit your, if you find something from your particular, not just bacteria, it can be anything. Now, you can deposit a genome, you can deposit a bacteria. It's like building a database. So this is deposited in GenBank under that particular accession. So if you just put that accession, you get the entire genome of SM8. And then our bioinformatician, he assembled it using a meta-assembler tool which is a software. All these are bioinformatics. Trust me, I had to learn some of it, but I do wet lab and dry lab, so we had to hire a bioinformatician for analyzing this then, and then it was annotated by a genome company, which is IMG, and we found out that there were 15 gene clusters, and 9 of them could be involved in antibiotic production. And of these 15 gene clusters, we found some are cryptic ones, which is like that can be developed, and that can be a novel drug as well, but we couldn't pursue further, as I mentioned. It's just that we didn't have funding to go forward. But then we have mentioned in my thesis, as well as in some of the papers, what those clusters could be. And some of the students had actually compared this genome with some other genome, and they have done some analysis, but I don't have data for that because it's their work. I don't want to present that. So as I said, this SM8 is kind of uh, very good strain to work on to get more antibiotics and, new antibiotics and antifungals. And this is what you can do in future. Like, you can just create a library, sequence the clones, and then also identify what antibiotic it is using NMR and so on. So this would have been my future work, actually, if I would have pursued that further. Um, so you can find my publications here. So these are different publications. So far I have been having, and I'm still actually two more, just not related to this, but what I was doing with the AMR projects and so on. And that's my ORCID ID, if anyone is interested. And yeah, that's my LinkedIn profile. You can contact me, you can email me. So that's my email ID, and that's my LinkedIn profile. And that's my Twitter account. If anybody wants to tweet, they can hashtag it. That's it. I'd be happy for questions. And sorry for the technical, because this laptop is quite old. Mine one is it's under repair, so I have to use my husband. So I'm sorry about that if it's crashed down a couple of times.
Yeah.
So.
Thanks very much, Lekha. So that was a great talk. I just for the audience, I told Lekha there'll be a mix of students and scientists, so I can see the effort she have put to to make it so basic that even the first year undergraduate students could understand. I'm pretty sure phylum class engines people would know. Okay, so before discussion, we would like to ask you to please give your feedback. The feedback link is in the chat box now, so please fill the feedback form, and Lekha would like to see what do you think about her talk. And we would also like to announce, if any of you are happy to volunteer for AFS Talks, please do let us know. We'd be interested to know in which ways you are willing to contribute developing AFS Talks as a knowledge sharing platform. There are a host of activities that we are planning, looking forward to deliver through AFS Talks. So please do not hesitate to contact us and discuss what value you can offer to our services. So going to the discussion part, we have 15 minutes for discussion. Please feel free to unmute yourself, which I will, yeah, I just allowed all of you to unmute yourself. So yeah, so if possible, also type your question in the chat box so everyone, including the speaker, can read and understand the questions. Any questions, please, you can ask. Thank you.
I think there is one question here by Shriya.
Yeah.
How to select a strain for MRSA as antibiotic resistant? How to select a strain? So basically, if you have now actually in a lab we do have an MRSA strain. MRSA is nothing but it's the Staph aureus, which is resistant to methicillin. So if it is resistant, if it shows resistant to methicillin, that would be called as an MRSA strain as well. And so that is an MRSA. So I have tested DSM-8 crude extract against MRSA, which I think the question is quite interesting. And it was showing that it can kill MRSA, but as I said, I don't know which antibiotic it would be from SM8. Have I answered your question, Shriya? Okay, perfect. Thank you.
Anyone else?
Now, in continuation to Shreya's question, actually, we do have another kind of resistant as well, vancomycin resistant, and that is also, it's called VRSA, but that is also with Staph aureus, you know. So that's why I said how antibiotic resistant strain is going to be a bit of alarming situation to the entire human race, because there are a lot of these becoming resistant now. So the Staph aureus resistant to vancomycin, it's called vancomycin resistant strains. So, you know, just like how TB, you have the I forgot the TB one. It's there's one thing which is kind of it is becoming resistant. So I forgot the antibiotic though, but those are the ones which, you know, we have to be careful. The Candida is becoming resistant as well. So basically, if you don't fight back, you know. Any other question?
Anyone? Yeah, just come forward, because we are all in same generation. So.
I would, okay, we have a question. So if you have to prepare a drug for MRSA, which species would be selected from it? It's difficult to say which one, because whichever is effective, you can select. So my main, like, if you read the literature, there are a lot of actinobacterias which have been proven to be good drug candidates. You know, any bacteria which comes under actinobacteria, as I said, the classification, actinobacteria, it can be strep, it can be micrococcus, all these are genus levels. They are found to be very good. But recently, I have read papers wherein bacillus strains from marine origin have proved to be, you know, resistant as well. So that's so you cannot pin down to one particular strain. As I said, it need not be streptomyces, it can be something else. And there are a lot of bacillus. If I'm right, there was pseudo-vibrio strains, which is I forgot the phylum now. On the top of my head, I'm not getting. It could be either Firmicutes or something else. But yeah, there are pseudo-vibrio strains which are showing wherein they have developed, you know, new antibiotics for MRSA. So these are all different strains. You cannot pinpoint which one. It all comes on, you know, on practice. Like you just have to screen for everything. Ashraf has asked a question. Have you attempted any peptides capable to combat? No, Ashraf, haven't. Good question. I have moved from my marine field. But I have written a grant on that though. Probably if I get that, I'll move to US and I might follow up that. But yes, it's a very interesting question, and hopefully around the world someone is doing that, because that would be interesting. Is it resistant to fungus, Rash? Yes, the strain what I was working, it's resistant to fungus. I didn't understand your question. Can you? Which is resistant to fungus? I didn't understand that particular question. Can you rephrase it for me?
Or you can unmute and
Yeah.
talk to Lekha. Rice. We can jump to the next one and then we'll come back.
Yeah. So the next one by Shamika is the compounds which you found. How did you test their effectiveness as an antibiotic or antifungal? So basically, the pre-screening, that's the pre-screening, which I just skipped because it will be in detail. So what you do is you put the strain, it's called an overlay assay. That's a very initial stage of my PhD. You put the strain in the middle of the plate, the media in which it grows, and then just top it up with your pathogenic strain, which has the solidified agar. And if you see a zone of inhibition, which means your strain is producing something to kill that particular fungus. So I have tested my against 5 fungus and 3 pathogenic bacteria. So the fungus would be Candidus albicans, um, Clebrata, Clevromyces, Streptomyces, Aspergillus, which is a pathogenic fungus. I've tested against all these 5 and also against E. coli, a pathogenic E. coli strain, MRSA in few of them, Staph, and Pseudomonas. So I have tested against a wide spectrum actually. Did I answer your question, Shamika? Perfect. Thank you. Royce, I can come back to your question, but if you can rephrase it a bit, it would be helpful. You asked, is it resistant to fungus? So I didn't understand which one are you talking about? Are you asking SM8 is resistant to fungus?
He said fungal infection.
Oh, fungal infection, fungus. So, for example, okay, I think I'll probably put it in simpler terms. So, for example, if, no problem, I can understand the network problem. I know the issues because I have friends from India as well who always have network issues whenever I talk. So, the one which I'll put it in simpler terms. So, for example, Aspergillus, okay, you have an Aspergillus infection, and Aspergillus they are very pathogenic fungi, and sometimes if the spores of it goes into the lung, you get lung infections. Now, if you're going to treat an Aspergillus infection, you will have to give an antifungal drug, okay, and if that antifungal drug, if that particular Aspergillus is resistant, that antifungal drug, whatever you're giving, is not going to work. Okay, that's when you call that particular Aspergillus as resistant to the drugs. That's when it's called that it's a superbug. You call it as superbugs or multi-drug resistant strains. Am I clear?
Not sure he's trying to speak.
I think he's doing. Okay. Yeah. Okay. Perfect.
Anyone else have a question?
And I just want to say, no question is stupid in science. Trust me. I have searched for stupid questions, so no question is stupid. Please be, open, because sometimes it would be challenging for me. In fact, it would make me think, to answer your question. So you can be just open.
Okay, I will ask something which is, I thought it would be stupid.
It's okay, Deepak.
How do you justify your intuition when you publish a paper?
Oh, that's interesting actually, because my first paper, for example, I really thought that oh, it's going to go through because my boss has looked into, and my intuition was like, oh, I'm going to get it, published and everything. But then the very next day, so it got uploaded in the system at about 9:30 or something. And then the very next day it came rejected because that didn't satisfy the scope of the journal, So I don't believe in intuitions for the journal articles. No, I trust only my work, not the journals.
You still have to find something to justify that.
Yeah, I mean,
Little secrets of the lab.
No, we did that. We actually looked into different other scopes wherein they would accept ecology papers, and it got accepted straight away. It's just that we selected a journal which was not right for that scope, you know. So even when that happens, I think, to all the scientists, basically, or all people in any field for that matter. If you don't know the scope of the journal and if you submit it, it can come back. But that's not a rejection, you know. It's like something you would think, okay, why not try somewhere else?
Yeah.
So,
That makes me happy now.
Ashraf has sent me a question. How many molecules you come across in fluorescence property? If so, how you utilized it? Fluorescence. I don't think I wouldn't say fluorescence, Ashraf. What I have come across is only like, it has, in the plate, it didn't show me fluorescence though. It just showed me a different color. So I think it was producing something else as well. I wouldn't say fluorescence. I haven't come across fluorescence. But I did some experiments with some other methodology, but that is completely different. So I haven't come personally across this fluorescence property, but it could be. There could be literature out stating, fluorescence property, but I haven't utilized it actually. I haven't used it. Have I answered your question?
I couldn't see that question because I think he put straight to you.
Yeah.
Yeah.
He put straight to me. Yeah.
It's pretty quiet today. 100 people, but no questions.
Either I could be a bad speaker that they didn't get it. It could go either way. So
I usually think sometimes think that way. When people don't ask questions, and then you start feeling, oh, maybe I was not clear about what I'm talking. but it's also people are maybe a bit hesitant to talk.
or maybe sleepy because it's around 8 o'clock and 8:30 is it in India. So
I should be the one sleeping because I'm on like 12 a.m. the night.
12 a.m. Yeah.
Okay, I don't think.
Any of the other hosts who have a question, because
There we have a few people on board. Maybe one of them or two of them or all of them can ask.
it would be interesting because I wouldn't mind answering questions from them as well.
Any questions, Mukta, Pradesh, or Sajina?
It's not a question, but it's a comment. This is the first time that I'm hearing this much deep about sponges and all. Even though I learned something on sponges when way long ago, and this is the first time that I can see that okay, there's a lot of potential there in that field. So thank you for the talk.
Thanks, Pradesh. Thank you.
By the way, I didn't know that there was so much microbial load in sponges. So maybe it's true, probably. I don't know how many people are actually working on sponges, with the microbial stuff.
Yeah, because we have around 3,000 to 4,000 just bacterial strains, and around 1,500 fungal strains isolated from sponges. So it's a hell of a lot of work to do there. So we do give certain strains to whenever they come. When the students come, they try to screen as well for different activities. It could be antiviral, anti-cancer, and even across the globe. In US, we have sent to someone to test for anti-cancer because we don't have the the cell lines and everything to test here, or we would need another lab. So we do send it across when our collaborators ask for it.
There's a whole bunch of audience here. Everyone is from aquatic science background, and also in my last 20 years of students, I never heard anyone working on sponges. So that makes your talk actually very interesting. Is that a question coming? Not sure. Anyway, maybe we'll wind up the talk then. It's already an hour from when we start. So thanks very much, Lekha.
Thank you.
It was a great pleasure to see you. Maybe after how many years? I don't know. We used to talk by messenger, but yeah, actually face to face after a very long time. But you look pretty much same. I have white hairs anyway. All right, thanks very much. And I appreciate everyone who actually attended this talk, and people from different time zones. So thanks very much for joining the talk, and we'll have another exciting speaker in 2 weeks' time. I hope Lekha can join sometime with us, if it interests your area of research. And I hope to get you again for, doing some other services, which we will discuss it anyway. Okay, so that's it. Thanks very much, everyone. Signing off. Yeah.
Thank you.
Bye-bye.